Hello and welcome everybody to another DecodeME webinar. I'm joined today by colleagues Chris Ponting and Andy Devereux-Cook. They'll be speaking in a moment, so I'm sure they'll say a little bit more to you all, so we won't take time with introductions now. But I do want to welcome those people who are watching on the webinar, but also those that have joined us online through Facebook. As always, we will record this webinar so that people can access it afterwards. Or if you need to pause at any point, you can come back and watch again when the video's live. And as always, we will have a transcript available to assist people if watching is a bit too much. So today we want to talk to you about our plans and the next steps, and also we will have an opportunity for questions. Colleagues Claire, Anne and Ella are all in the background, ready to take questions and feed them through and deal with any issues with the technology should they arise. So thank you to colleagues for helping us with that. So I'm just going to share some slides with you. So just bear with me a moment while we pull these up. So today we're going to talk to you about the DecodeME study extension, and we know that that has been a disappointment and frustration. We feel the same, so we want to share with you a little bit more information about that. We're going to give you an update in terms of where we are with the project, and I can honestly say, hand on heart, the whole team has been working incredibly hard in the background. That includes our PPI, patient public involvement members, people with lived experience, the team at Edinburgh, and also the team at Action for ME and others that we're working with. We're then going to say a little bit about what we've got coming up over the next few months, and then we'll finish with some questions and answers. So as many of you will know, we were originally due to complete the DecodeME study by August this year. Throughout the project, and we've talked to you previously about some of the challenges that we've had that affected the timeline. So things like Amazon buying up all the boxes during the pandemic and lockdown, which impacted on us. We've had postal strikes, and we've had changes in the people that have been working with us in what's called the supply chain. However, we've managed to overcome those, but the one challenge that we weren't able to overcome were the operational and capacity issues at the UK Biocentre, and they've been the most significant factor to affect our project completion timeline. While we've been able to mitigate some of the delays, and we've worked incredibly hard with them, we've been in touch every single week, sometimes every single day, working with them to try and overcome the issues. We have not yet got all the data that we expected to have back from them at this point. So when people send their samples in, they go to the centre, they're processed, and we get the data back at Edinburgh University. And we've not yet got all of that data back. And I must say, I want to reassure you, nothing is going to happen with the samples that have been sent in. We will get the data. It's just taking longer than we had expected. And we now need more time in order to quality check and analyse the genetic data. And there's still quite a lot to be done. The people that have taken part in the study, completed questionnaires, sent in samples, have done an amazing job. But the next step, the extraction of that DNA from the saliva samples at the Biocentre, is what has really caused those delays. So we don't have enough time to perform all the analysis that we needed to by 2024. And that's where we then approached our funders, who agreed to give us an extension, and they have funded us for another year to enable us to do the work that we need to. We've discussed this situation with our scientific advisory board as well as our funders, and they also agreed with our assessment, and that should give us sufficient time to finish the project. I can honestly say, lots of honesty here, and you'll hear this from Chris and also from Andy. People are working really hard and are committed to completing this research to the highest possible scientific standards, ensuring that our findings are as robust as possible. We all know in the community that there have been many false starts, and we are dedicated and committed to making sure that our findings are scientifically valid. Our funders have given us the extra money, as we've said, so that we've got the team in place, and there's quite a large team, and they're having to work really hard with quite a lot to do. But we will ensure that we deliver as soon as we can and give results as soon as we can. But we will only do that when they reach the standard required. So we are now working to a deadline of August 2025. And as I said, we will release results as soon as possible before this date. So I'm now going to hand over to Chris, who is going to tell you a little bit more about the work that we're actually doing at the moment.
Thank you, Sonia. So participants who provided DNA will last remember that you've sent your samples to the UK Biocentre. And that is, for us, the beginning. There are so many steps after that, but for you, you probably don't know so much about what happens afterwards. So I just thought I would go through each of the steps in turn, and that perhaps helps to understand the complexity of the operation and what has caused the delays. So when the samples arrive at the UK Biocentre, the sample is split into two, and the second sample is kept for future research. So the important thing is to say that each sample is incredibly valuable to us, and we've managed to get over to Biocentre how valuable each sample is by going to visit with members of the PPI team. So the Biocentre takes receipt of each sample. It associates a barcode with a sample, which allows the tracking of the samples going right from that point until the end when we do the analysis. The amount of DNA in each sample then needs to be measured. And after that, the extraction occurs, and there is the replating, so the taking of a portion of that sample and putting into a plate that with other samples then goes to Thermo Fisher here in the second step for genotyping. So before we get on to that, because of the delays that we've been discussing, so what's not gone to plan? Our plans have been very extensive. We have been planning for many years, but we didn't plan for this. So what's been going wrong is that at times the samples have not been receipted properly when they turned up at the door. At times the tracking of barcodes hasn't been done to the highest standards, and not all of the quantification of the DNA was done perfectly. So there have been, unfortunately, errors and inconsistencies in the process that have caused us to meet with Biocentre twice a week and to go through literally sample by sample what has happened for each one of them. Biocentre have engaged in that process very well indeed, and we have to thank them for that. They have produced a 62-page report about all of the different samples and what of the issues that have gone on and all of their checks that they have now employed. And we're signing that off soon. But the first version of that report goes back to last summer. So you can tell that I hope you can tell we've been working hard on that every week for those two meetings, and holding to account the organization that has been doing all of the work. This has not been something that we have been able to do and to mitigate. The MRC, when they gave us the extra money, they said that their panel agreed that the major delays had been outside of the control of the principal investigator, ourselves, and were being actively managed by ourselves. So that's what they said. So the first set of plates with the DNA samples have gone to Thermo Fisher, and the first plates have come back, the data has come back to us, and that's the third step in the process here, where we compare the data with UK Biobank controls. Thank you. And we've done that for the first batch. We last week got the second set of data, the second batch, and we're looking through that now, making sure that it's of high quality. It does look like it's of high quality. There'll be 2 more of those batches, which will come really quickly, because I'm pleased to tell you that there is no backlog of DNA extractions. Extractions are almost complete, but of course we're still meeting twice a week to ensure that that happens. So by the summer, we'll have all of the data, and we'll do the data cleaning, what's called quality control, and we'll be comparing it with the DNA that comes from the UK Biobank, which represents healthy controls. So the comparison, we've discussed this in previous webinars, allows us to say where in the genome there are letter changes that are more frequent among people with ME than those in the general population, which will indicate where in the genome there are risk factors for ME, which hopefully will tell us what are the systems that are going wrong in people with ME. It's the nervous system, the immune system, mitochondria, autonomic system, etc. So that's then will allow us to put together all of the results, and we will write them up as soon as we can with respect to us knowing that we have done the best possible job and we've done all the checks and balances, and then we'll write them up for peer review. Thank you. Next slide. So whatever we find, we have to make sure it's robust. We'll do everything we can in-house, but there is an additional way that we can ensure that what we have found is correct and right and true, and that is to collaborate with others on a replication of the finding. So ahead of time, and we're announcing this today, we are collaborating with Hanna Olila and Anina Turvi from FinGen in Finland, who already are analyzing ME data from electronic health records from different sources from Finland, FinGen, from the USA, the Mass General Brigham Biobank, and the Estonian Biobank. So this then becomes an international effort using thousands of people's data with DecodeME to ensure that we together internationally have the best result for the community as possible. Good. Next slide. So what then? It isn't an academic exercise. What we're doing is to try and accelerate research towards clinical trials and new drug targets as fast as we can. So we're up in the top left, we're doing the genetics, and we're trying to find these hits, which are the red circles, that will lead us to understanding what's going wrong in people with ME, and how the different genes and variants relate to one another in a disease network. That will help to potentially anyway repurpose drugs, if there are any. We'll compare to known drugs and databases, and from that there will be an opportunity to test drugs in trials on cases and controls. Now, we of course are only in the top left-hand corner of this graph at the moment, but we're planning ahead, and so as many people know, we are already working with Precision Life, it's an artificial intelligence company, and who have partners in pharma. So that will then provide an acceleration of anything that we provide, we produce from DecodeME into the right fields towards new drugs and new drug targets. So I think I'll pass over now in the next slide to Andy, who's going to finish the presentation.
Thank you, Chris and Sonia. So I'm here to finish up our slides. As you can see, we've got a project update on the numbers who have taken part. So we started with almost 38,000 people who signed up to the study, created an account with us. Just under 27,000 of you completed the first questionnaire. And just that part alone provided a lot of valuable data, which will be of use moving forward. From those, just under 22,000 were invited to provide DNA. Just over 18,000 of you have returned those DNA kits. And the newest figure is that of those just under 27K people who completed the first questionnaire, just over 15,000 completed the second questionnaire. So all of that is a fantastic result. So as a patient myself, you know, I can be tempted to look at those figures, particularly the top line, and think, well, that's a large number, but then we're losing numbers as we go down. That is something that happens in studies. You know, for various reasons, people do not take part in the next stage, which obviously is their choice. And even to get to the second questionnaire, where over half of you were willing to take part in that, just shows what a fabulous contribution you've all made to DecodeME. So thank you all for your efforts. I know personally the cost in energy and time that takes. So if we move on to the next, thank you. Next steps, we've already kind of covered it. We're going to obviously continue to send those batches of DNA to Thermo Fisher for genotyping, get that genetic data back, do all the necessary analysis, which is more easily said than done. It takes far more time to actually do that analysis than obviously to say that sentence. Publish the results eventually. Oh, that will come, well, ultimately when it's ready. There is a great awareness throughout the team that we would like to do this as quickly as possible, but balanced against that is the quality of our final output. And ultimately, within the time limits that we have, we're aiming for the quality of that final output, because that will provide a starting point and a continuation for further research in the future. As you can see on the next line, next month we are going to be publicly opening up our access process for the data, obviously for what we've got already, and then obviously what will come in the future. So that process will allow other researchers to apply to us for access to that data to form the basis for future research from them. And then we're going to continue working on plans to build longevity as much as we can for the DecodeME cohort. Basically looking forward again to provide value to the community for not only for the money given to us by the funders, but again, the energy and time that you have invested in us. And I think that wraps that up.
Thank you, Chris, and thank you, Andy. We've had lots of questions coming in. I've got questions across a number of screens and in front of me. So if I'm looking away or typing something to colleagues, please don't think I'm not paying attention. We want to try and get through as many questions as possible. The first questions that I wanted to pick up, my computer is doing really funny things here. sorry. The first few questions that I wanted to pick up related to what you were saying, Chris, about some of the delays. And I think it'd be really helpful if you could just say a little bit about the DNA samples. People are concerned that they might have been lost due to the issues at the Biocentre, and worried that if there have been issues with those, that people are going to be asked to provide another DNA sample. So could you possibly just say a little bit more about the situation there for people?
Thank you, Sonia. So where samples were lost, and there were a couple of hundred that were lost, we have gone back and asked people for a second sample. We were anyway asking people for second samples because sometimes the sample that's provided doesn't process for a whole variety of reasons. And so when that happened and there wasn't enough DNA, for example, then we were asking people for a second sample. So all of that was happening behind the scenes. I don't have the numbers in front of me, but that was happening all the way through, and we made sure that people could be given the opportunity of giving a second sample, because our value is that every single sample is incredibly important. So we took that as a value right from the beginning, and then we followed it through all the way through. So there are cases where downstream of that, the sample is not able to be taken forward all the way down the process to the analysis. For example, where the DNA letter changes are read out, sometimes those are not taken forward. Those are minor. 99.8% of people, or 99% of people, I don't know what the number is, that will not happen. And so you can be reassured that your sample has been treated incredibly well from receipt to data analysis. And we've done all that we can to ensure that the value that it has translate into scientific value.
Great, thank you, Chris. We've got quite a few questions around the questionnaires. They are now closed. Both the first and the second questionnaire have closed. So I'm really sorry that if you're partway through or you missed the opportunity, really grateful. Our numbers are brilliant. So we are really pleased with the responses that we've had, and we don't want anybody to worry if they've missed out on that. We are delighted with the numbers that we've got. And we've had a question actually about the numbers, which I'm going to give to you, Andy, which asks, what numbers were we aiming for with this study?
As many as possible. The challenge, one of the many challenges for DecodeME as a study is that it was the first time anything on this scale had ever been done. So there were so many aspects of it where we had to take a best guess as to what was possible to be achieved. And to a large extent, they were achieved or they were, they were smashed by the community. So I was mentioning earlier about how, there was it could be perceived there to be a drop-off between the numbers who originally signed up and then who answered the first questionnaire, and the numbers were getting smaller down every time. Like I say, that happens with basically every research study out there. So, but the numbers that continued to take part, who took that next step that we asked them to, or gave them the opportunity to, rank right up there with the best results from other studies from other fields. So the numbers that we were aiming for were our best guess about what was possible with the community, with what was also required for the scientific basis of the study. And there's always more is better, more or less. And we're absolutely delighted with the numbers that we did eventually get.
Thank you, Andy. I'm just going through some of the questions again. Just a reminder that we've now closed to both the DNA samples and the questionnaires. So please don't worry, you haven't said them. We've had quite a few comments around that. Also a question around whether or not the study is going to be available to people in the US. This is a UK only study and we've now closed recruitment. But as Chris shared, we are trying really hard to work with researchers in other countries so that we can extend this study and hopefully replicate and validate any findings. We've had some questions around transcripts and slides. Everything will be available afterwards. Do give us a little bit of time to get the film ready and this transcript sorted, but we will make sure those are up on the website and available for you to be able to use afterwards. There's a question here, Chris, about the lost samples. Were they destroyed?
Where there was the barcodes, the fidelity of the barcode tracing was lost, we've destroyed those samples because we would not know from whom they came. But as I said previously, we have gone back to those whose samples were affected, and we've asked for second samples where possible.
Thank you, Chris. We've got quite a few number of questions about drug testing and will low dose naltrexone and others be used. We don't know. Our focus is on getting the genetic data. And as Chris shared, we're working with others to ensure that our data can be used. Chris, I don't know whether you want to add anything to that.
I just want to thank people for putting down in the second questionnaire all of the treatments that that you've tried and saying whether you felt that they were helpful or not, and that's going to be incredible data to pour through, and we're going to do that as soon as we can.
Right, thank you. We've got a few questions around data security. So can you say a little bit more about how the samples are anonymized and genetic data is secured and who the data is going to be shared with and what data will be shared? Sorry, I'm trying to condense questions.
Yeah. So everything is in the participant information sheet that everyone will have seen at the beginning of their journey. And essentially, you know, we have asked people whether or not you wish to consent for sharing your data, your DNA, whatever, and we've said quite openly, if you don't want to do that, then that's absolutely fine. For those people who have, we take everything that you've given us very seriously as to its data security. So we've gone around the houses with respect to ensuring that your data is held securely here. We don't share personally identifiable information except with the company that sent you the spit kit if you got one. And when we shared that, we did so under a contract where it was clear that everything was held securely and data, that personally identifiable information, would be removed from their systems. So this is a very high standard that we've adhered to, that every project like ours adheres to, and the University of Edinburgh, as a data processor, treats very seriously, and we've had to go through internal processes to ensure that we are held to those high standards that all of you deserve.
Thank you. We've got a question here again. I think this is going to be for you, Chris. I understand you previously discussed this research in terms of medication. I wanted to know, does DNA affect our nervous system? And does anyone requested to access this research in reference to the nervous system and current theories around nervous system dysregulation?
But the great thing about the genetic study that we're doing is that it will answer the question, what is going wrong, whether it's the nervous system, the immune system, or whatever system. If it is the nervous system, then indeed we know from other disorders that the nervous system can be changed by DNA letter changes. And And so that's the reason, like, most of our studies like this, we are doing genetic study, is that we'll be able to find out if it is a nervous system, what cells are going wrong in a nervous system disease. If it's an immune system, exactly the same. What cells are going wrong? What are they the cells only that have been challenged because of an infection, like a virus? And that will allow not just us, but people worldwide to drill down with their own expertise on what are the minutiae of what's going wrong with respect to molecules and cells. And so this is the launch pad for new research into the future, whether it's nervous system or not.
Thank you, Chris. Still more questions around the questionnaires. We're not giving results of the second questionnaires individually, but we are hoping to publish, and we will do that when we have the results, we've analysed them, and we're convinced of the robustness of them. And we check and check and check, and we will publish those, and we will make them available as soon as we can. We had a question saying, will the results be put to the GMA? I think that might be the General Medical Council. Obviously, once the results are available, we share that. We've had another comment saying that these webinars are a bit wordy. I'm sorry that they're not as accessible for you as maybe others. We want to get as much information in as possible, and we do recognise that that's not okay for everyone. We try and ensure that our blogs, we have a quick read and then more information. So we do try really hard, and I have to say that's the massive value of having people with lived experience working with us as part of the team. We try very hard to make everything as accessible as possible, but we do recognise there will always be gaps, and I can only say we're very sorry that we're not able to make this less wordy for you, but the transcript will be available. You can take it in chunks. We've got questions here. A question, what about if ME patients do not have any genetic variation that makes us prone to the illness, and that it is a result of, e.g., virus infection plus failure to rest? Would that show up, Chris?
Yeah, so everything that we're prone to is really a spectrum. And it's your environment through infection, etc., that will push you towards being at greater risk. It'll be DNA letters that will push you to less risk or more risk. It's the combination of those that plays out as to whether someone will have an ME diagnosis or not, rather than being, do you have a gene that means you'll get ME? It's not as simple as that. This is a complex disorder. And we absolutely know that 2/3 of people have an infection prior to ME onset. And that's telling us something, isn't it? This is telling us that infection has meant that it's the environment that contributes hugely to people's risk of ME. Why we're looking at DNA is that it's the DNA that can tell us what's going wrong, not that the DNA is telling us that it's the major component of what's going wrong.
Thank you, Chris. I'm very conscious we haven't got much time left, and there are quite a lot of questions. Just a reminder to people, we can't answer questions about your individual circumstances, when you got ill, why you got ill. So I apologise if you've taken energy to give us a question, but I hope you understand that that's not something we're focused on in this particular study or webinar. Andy, do you expect or anticipate that the study will result in the reclassification of CFS-ME from the current classification as a neurological disorder to something else?
I think it all comes down to what our results demonstrate. I have to admit that I'm not up on the exact processes for how something is determined to be defined as a neurological disease and how it's then defined as something different. But so I can't really give a greatly informed answer, but and so my answer is unfortunately going back to it will depend on the results, which isn't that helpful. so apologies for that, but I think that that's where it is. That will be how we determine that once we know what we found out.
Thank you, Andy. We've got lots of questions around how we might stratify the DNA, look at it from different perspectives, look at gender, look at age, those kinds of things. We're absolutely going to be doing that. Lots of lovely comments from people. Thank you for feeding those back. We really do value your feedback, good and bad. Question here, is it possible that ME/CFS is an umbrella term for a number of different illnesses sharing the same symptoms? And could it be that long COVID, fibromyalgia, etc., are actually ME? And we've had a sort of a linked question around FMD as well. So Chris, can I maybe ask you to take that in the first instance?
Thank you, and I apologize for being too wordy earlier. About half of people with long COVID have symptoms that match the symptoms of ME. So there is an overlap, but it's not the same. And that means that we should know that there are commonalities, but there are also differences. And that's exactly how we will treat it scientifically.
Thank you. We've got questions here around the blood-brain barrier. Can I ask whether this research would show any anomalies within the hippocampus or other parts of the brain that controls the immune function response? Chris? If
there is a genetic signal in a gene that does that controls the immune system from the hippocampus, then that hypothesis will be supported. There will be 1,000 or more similar hypotheses that could yet be supported.
What about international replication? Is the methodology being replicated in Finland and USA to ensure standardization?
It will not be the same because the people, their DNA is associated with their electronic health records, not a diagnosis of ME. But because the electronic health records should give us a good indication of whether what we are seeing with precise ME is also occurring for people with more general post-viral fatigue, which is how the electronic health records record anyone with ME, but others too. Sorry, that's complicated.
And some of these questions are complicated, and I think they're brilliant. You know, the engagement that we've had from people in the community is really good. And I have to say, when we've had some dark moments, when things are going wrong and we've got huge hurdles, your support and your engagement has made a massive difference to us. So I'm afraid that's we're coming to the end of the webinar now. And I just want to say thank you to Chris and to Andy, but also all of the people working in the background. They, as I said at the beginning, they've worked incredibly hard and are very dedicated and committed. And the three of us tend to be the face of the study, but actually there are many more faces in the background. We've had a question about how many people that participated in the study represent the UK population. If we work on the basis of 250,000 people in the UK with ME, we all know it's a very old figure and outdated, then around 1/10 of that community have participated in this study. I think that is phenomenal, and we should really congratulate that. So just to say that we will look at the remaining questions. We've got through a lot, but if there are any questions we think we can answer and put up onto the FAQ, on the website www.decodeme.org.uk, then we will do. There will be more webinars, there will be more blogs, and you can access the film of this and the transcript, hopefully in the next week or two on our website. Thank you for your time and your energy.